Now it's my pleasure to introduce Brandon Cooper, our webinar speaker today. Brandon's an associate professor in molecular and biomedical sciences at the University of Montana. Brandon's an evolutionary geneticist broadly interested in population biology and endosymbiosis. Most of Brandon's work focuses on explaining the spread and maintenance of endosymbiotic Wolbachia infections in insects. But he has broader interests, and his work includes work on host speciation, and more recently, he's even worked on the evolution of the SARS-CoV-2 virus. Brandon, it's great to have you with us today and for you to share your research with us. At this point, I'm going to turn the floor over to you. Thanks again for being here.
Okay, well, a big thanks to Dave and his team. I've really enjoyed interacting with all of you and preparing for this. I'm really looking forward to it. Also, thanks to the wonderful speakers in this series, and thanks to all of you for being here today. As Dave noted, I'm an associate professor at the University of Montana in Missoula. As he also noted, I'm more specifically an evolutionary geneticist and a population biologist with broad interest in biodiversity. Like many of you, I'm obviously also interested in the diversity of microbes that associate with hosts. This includes a diversity of endosymbionts living inside host bodies, some of those examples here in the systems I'm showing on this slide, with such associations being particularly common in insects. Obviously, we're all here today because of one of these endosymbionts, Wolbachia. My primary interest in Wolbachia in terms of the basic biology is the fact that they're the most common known endosymbionts in nature. I think interestingly, they're not restricted in their distribution within insects, so they infect all major insect orders. This is in addition to other arthropods and some nematodes. So some of what I present here at the beginning will be a little overlapping with the past speakers, but I want to give a little bit of an introduction just for people that may not have seen the prior two. So I think the discovery of Wolbachia is interesting because the story starts with population biology. And as I said, I'm a population biologist. And it was on the islands of the South Pacific in the late 1880s. And specifically in 1878 in Borneo, collectors could not find males of the blue moon butterfly shown here. And then in the 1920s on Fiji, Simmons, who's an entomologist, discovered 100-to-1 female-male sex ratio. So for any of the evolutionary biologists in the audience, this is obviously surprising. And then if you were here last week, you would know that later on, Samuel Wolbach discovered a bacteria that resided in the germline of the mosquito. And it turns out that this is the endosymbiotic bacteria that we now call Wolbachia based on that discovery. So what are Wolbachia? These are maternally transmitted Alphaproteobacteria. What I'm showing you here is a developing female oocyte. And you can see here Wolbachia being the little stain puncta inside this oocyte. And this oocyte is at a particular stage of development where Wolbachia has some work to do to get into the germline, which we'll talk about a little bit later. We often think about Wolbachia in the reproductive tract for a variety of reasons that I'll talk about. But I do want to note that they tend to be distributed throughout host tissues. So I'm showing you an example here with Wolbachia in blue in a host brain, and then again, Wolbachia in the developing female oocyte reproductive tissues. Now, where Wolbachia are, the particular abundances, when they get there, that can vary a lot among systems, but on average, we see Wolbachia in the soma and in the reproductive tissues. Their genome's about 1.2 megabases. It's pretty well characterized, at least for model variants. We have a lot of good genomes at this point. There are some atypical varieties of Wolbachia genomes that are smaller, but on average, they tend to be about 1.2 megabases. And that includes some prophage regions and other mobile elements, which we'll talk about and which have been talked about a bit in this series so far. If you're aware of Wolbachia, it probably is due to a couple of reasons. I think the first reason they became particularly recognized and particularly exciting to biologists like me is that they manipulate host reproduction to favor their maternal transmission. So remember, moms pass on Wolbachia. So you can think about the genetics like you think about, say, mitochondria. And Wolbachia has evolved these nice strategies to get into the next generation. So some of them can induce parthenogenesis. Some of them can feminize males. So if you can't get into the next generation through a male, you can feminize a male to get into the next generation. As I introduced with the original observation of a phenotype that we now know is due to Wolbachia, some of them can kill males. The idea here would be that if you kill male embryos, mom can devote more energy, say, to females, to the female embryos that carry Wolbachia, right, and can pass them on. And then, of course, one that was a focus last week, cytoplasmic incompatibility, which is also a trait that we work on. I will cover what CI is in terms of the phenotype. If you're interested in the very specific mechanisms, I would urge you to look at the video of Dr. Carr's presentation from last week that really covered the details of the mechanism. So I won't get too much into the mechanisms today. But I will cover what CI is because it is important, because it provides a relative fitness advantage to infected females. And so if you're looking at these crosses, the unfilled circles here, sex symbols, are uninfected individuals, so individuals not carrying Wolbachia. The filled symbols are individuals carrying Wolbachia. If you're an uninfected female and you mate with an infected male, everything's groovy. If you mate with an infected male, the filled sex symbol here, you get reductions in embryo mortality. Importantly, you can get rescue when the female's infected with the same variant or a very closely related variant. And so practically speaking, what that enables is that infected females can mate with both classes of males, and that obviously can provide them with a relative fitness advantage. I do want to note, because I think it's important, in particular from my point of view on the population biology, that CI has no effect on initial spread of Wolbachia from low frequencies. You can imagine that Wolbachia gets into a new system. In that moment, there's very low infection frequency. So uninfected females in the populations tend, just based on probability, to mate with uninfected males. So we know that Wolbachia must initially spread through host populations via other ways by positively affecting host fitness. I'm happy to talk about that. It won't be a focus of this talk, but just know that there's some evidence of how they do that, but it's a pretty big gap in knowledge in most systems about how Wolbachia are positively affecting host fitness to initially spread. But what happens is once they become relatively common, CI kicks in, and that's the moment where you have Wolbachia at a high enough frequency such that uninfected females now start mating with infected males. Those infected males start hammering their embryos, and that relative fitness advantage shoots Wolbachia up to higher frequencies. So a signature of a strong CI system is you go out into nature and you look at the population, and what you see is that Wolbachia are very common. So I'm showing you a classic case here of this variant we call WRI, based on its discovery in the 1980s in Riverside, California, in Drosophila simulans by Ari Hoffman and Michael Torelli. And here I'm showing you WRI abundance in Australia in 2012, with the piece of the pie that's red being the proportion of hosts that are infected. These frequencies now at WRI are pretty stable globally. About anywhere you go, they're about 93%. You might ask why they aren't fixing. So why are all individuals not infected? And that's because Wolbachia tend to be imperfectly transmitted. So infected moms don't get Wolbachia into all eggs. So the frequency that you find Wolbachia at in nature it's some balance between fitness effects, effects like CI, and then maternal transmission, each generation introducing uninfected individuals. Okay, so strong CI drives Wolbachia to high frequencies. CI is obviously important for both pest and controlling pest populations and vectors that can cause human disease. This comes in a couple flavors, as you've seen in the prior talks. I'll briefly cover them here. One is a population suppression approach. So you can take a CI causing Wolbachia and you can get it into a species like Aedes aegypti. You can then release males that carry that in mass. Those males mate with the naturally uninfected females. Their eggs don't hatch, and you get reductions in the population size. And this is being carried out in various places with some success. One caveat is this requires an ongoing male releases. So this would be a strategy where you would go release males to potentially deal with some outbreak or some pest population, reduce the population size, and then once you stop doing those releases, you would get rebound in the population size, presumably under most situations. Another is population replacement. And so this is a strategy primarily led by the World Mosquito Program and Scott O'Neill, with contributions from lots of other researchers in terms of the basic biology that feeds into this. Scott and the World Mosquito Program are having a lot of success. There are a bunch of sites across the globe now where they're releasing Wolbachia-infected mosquitoes. The difference in this strategy is you're actually trying to establish Wolbachia like W. mel from Drosophila melanogaster into these populations, because that variant and some others are pathogen blocking. So they have this characteristic where if a mosquito is infected, they're less likely to pass on things like dengue, diseases like dengue and Zika and others. And so this is different in that you want to establish these, and then you could presumably walk away and you now have an infected population that block these pathogens. Again, CI won't enable you to spread these initially, so there's a period of human releases that get them to an appreciable frequency, and then after that point, CI helps maintain these pathogen blocking wall variants at a high frequency. That frequency, again, is determined by imperfect transmission rates. The more leaky transmission is, the lower that equilibrium frequency will be. This strategy is having success, and so there's now data coming out showing that where these releases are happening, you're getting reductions in disease. Notably, this is projected to protect a lot of people in coming years, with projections last I look of hoping to protect 500 million individuals by 2030. But we work on the basic biology and questions that I think inform these strategies. And as I was trying to think about what I might talk about today that this group would be interested in, I settled on two questions. And one of those is among species spread. So thinking about how Wolbachia infiltrate new host lineages to become common. I've told you they're the most common known endosymbionts in nature, infecting these various insect orders. And so we're interested in how that happens. And then also within species prevalence. So how common are Wolbachia in different systems? I noted that in WRI, it's about 93% and stable globally, but you'll see that in other systems, it's much more complex. And so I'm going to focus on these two today. So let's start with among species spread. And a basic question about how do Wolbachia infiltrate host lineages? So when we look out across insects, we know that at least half, more than half, are infected with Wolbachia. Again, this is in addition to other arthropods and some nematodes. I think I consider that to be a lower bound because there are many systems where only a few individuals have been sampled. And so if Wolbachia infection frequencies are relatively low, you wouldn't have picked those up by the PCR analyses. There are other cases where Wolbachia titer in host tissues can be low and you don't always pick up those infections. So I think about half, more than half infected is a lower bound. So how did they get all these? How did all these species get infected? And I think a plausible hypothesis, at least about 10 years ago when we started thinking about this, is that hosts probably acquire their Wolbachia on average from common ancestors during speciation. So the idea here would be that Wolbachia has gotten into the tree, infected individuals back in the day, and then as hosts diverge, become reproductively isolated, those Wolbachia that they carry also diverge, and then we come back and we sample them, and you have sister species with sister Wolbachia that have been co-diverging with them. Just to be clear about this, what we could imagine is we have an ancestral population of insect hosts that are infected with Wolbachia. Think about this just like we would as the process of speciation and the evolution of reproductive isolation. Perhaps a physical barrier subdivides this population. In that moment, on either sides of this barrier, you have the same, on average, genetic variation in both host and Wolbachia. But through time, evolutionary process can play out. Host and Wolbachia diverge. We come back and sample these, and now we have two infected sister species. Both carry Wolbachia that their common ancestor had. They're now different Wolbachia that we would give different names, but they've been co-diverging for a similar amount of time. So how could we test if this is sort of the process by which Wolbachia became so common, that they got into insects and other organisms a long time ago and have been co-diverging? The way that we've been doing this is taking phylogenomic analyses of sister species. So briefly, the way that would work, you could go and you could sequence a bunch of your favorite hosts, making sure that you have sister species. So you could go into some clade and sequence their genomes. Fortunately, when you do this, you usually get the endosymbionts for free, or you can optimize your approach to make sure you get particular ones like Wolbachia. You can then do phylogenetics and produce what we would call phylograms to get these phylogenetic trees. And then you can start to test some hypotheses. So one thing you expect if Wolbachia are acquiring them in the way that I outlined, this process of what we call cladogenic acquisition or acquisition from common ancestors, is that you would expect there to be a shared topology. Okay, so here you have sister hosts that are infected with Wolbachia that are also sisters. So that's one prediction. And then another prediction would be that there's shared divergence time. So not only do you have shared topology, but Wolbachia have also been there since back in the day and codiverging with their hosts. So this has been, patterns of this have been evaluated going back into the 90s when Scott O'Neill in 92 looked at patterns related to this. And then Jack Warren's lab did some work in the 2000s. Our first work on this really marching through clades was in 2017, 2018, where we looked at these relationships across 50 million years of host evolution. So that tree I was showing you was one of our trees from this paper. This is across Drosophila species, specifically the melanogaster group of flies, diverged up to 50 million years. We have these various sister species. We then sequence their Wolbachia and we test the hypothesis as I've sort of outlined here on the prior slide. All you need to note is, so we have sequenced multiple Wolbachia variants from these different ones. So there are multiple individuals, multiple variants on the tips here. But the only thing you really need to notice is that it's a bit of a mess. Sister Wolbachia need not come from sister hosts. So we don't see a pattern of shared topology. And more than that, what we see are we have these hosts that are diverged tens of millions of years, but they're infected by Wolbachia that are diverged only thousands of years. And indeed, for some of these, the confidence intervals overlap with essentially this morning. So Wolbachia are getting into very divergent hosts on very short timescales. And we observed this in Drosophila, and this is specifically in this case, WRI variant spreading. So the one that was first discovered in Drosophila simulans. From these analyses, we know that it was originally donated from a species called Drosophila ananasi due to the paraphyletic relationship we see in this tree. Okay, so this is a pattern of rapid host switching of Wolbachia moving among very diverged hosts on short evolutionary timescales. We now know that this is happening even more regularly and among more divergent hosts. So we see essentially the same Wolbachia variants and host species diverged 350 million years now. So Hymenoptera and Diptera sharing Wolbachia. So this pattern we think is quite common. I don't mean to imply that we have no evidence of the cladogenic acquisition of Wolbachia. We have a few, but it's very rare. And about everything we look at, we tend to see divergent hosts that have very similar Wolbachia. One other pattern we see among sister species is we see patterns of intraggressive transfer. So in sister species that hybridize naturally in nature, sometimes Wolbachia move across species barriers that way, in the same way that you often, when you look at mitochondria, see a pattern of hybridization and introgression with mitochondria moving across species barriers due to those two relatively closely related hosts hybridizing. So I think what's interesting here is that Wolbachia are naturally moving horizontally, non-sexually among hosts more diverged than our Drosophila 80s, usually the donor and recipient of Wolbachia for some of these strategies that we've noted. So these are phylogenomic results. You might be wondering, okay, well, how is this happening? These are vertically transmitted endosymbionts that are obligately intracellular from the Wolbachia side. The answer is we don't know. There's some hypotheses. I think most of those that are most plausible center around species interactions. So you can think about interactions between parasitoid wasps and their prey. You could think about things like cannibalism, different species developing in the same sort of fruit substrates for different types of flies, for example. One thing we do know from nice molecular biology is this is possible from work of Horatio Friedman and others that Wolbachia can get into the germline and become vertically transmitted from the soma. But I think one of the interesting things moving forward is better understanding the mechanisms of how this non-sexual transfer is happening. For the rest of the talk, I want to focus on within species prevalence. So we're thinking about, we now know that Wolbachia are regularly moving into new hosts on short evolutionary timescales. So they're finding themselves in new backgrounds. And so I'm interested in how they spread in those systems, and then ultimately how common they are and what governs their prevalence in populations. So what can affect how common Wolbachia are, because not only are they common among species, but you see them at various frequencies within host populations, from both common to relatively uncommon. And one of the things I want to show you first is that it can vary a lot through space and time. So Tim Wheeler in my lab and others did some work in spittlebug showing that Wolbachia frequencies can vary over just only a few kilometers in two sister spittlebugs. They also vary among years. So in this system, we see variation through both time and space. Work by me prior and by Mike Haig more recently in West Africa on the island of Sao Tome, and from samples from the nearby island of Bioko, and then also continental Africa, we've noticed variation in Wolbachia prevalence in these other Drosophila species. So these are Drosophila yakuba and Drosophila santomea. They're two of the three species in the Drosophila yakuba clade and the Drosophila melanogaster subgroup. What I'm showing you on the x-axis here are the years at which we sampled flies, or the flies were sampled. And then on the y-axis, I'm showing you the prevalence of Wolbachia. So how many of those hosts, what proportion were infected? You can see for W. yak, a variant in Drosophila yakuba in 2001, frequencies were relatively low, 40% say, and then they increased in 2009, 2015, 2018, they look relatively stable. In the sister species, Drosophila santomea, which is an endemic fly that lives only at high altitudes on the island of Sao Tome, this tropical island. In the initial sampling, frequencies were around 70 to 75%. There are then years where they're sporadically lower, where only about 30% of the individuals are infected with Wolbachia. And then a few years later, it's back up to 75%. So lots of evidence of Wolbachia prevalence in host populations varying. Notably, we know from later work that I've done that the Wolbachia infecting these groups, work that I've done and others have done, that Wolbachia infecting these groups are what we would call WMEL-like Wolbachia. So the WMEL variant from Drosophila melanogaster that everybody knows and loves and is relevant for the pathogen blocking work. These are Wolbachia that diverged from WMEL only a few thousand years ago. So WMEL got into this clade horizontally. And then since getting in there, we also know that it's moved around horizontally between these species. One other interesting point is that at the same time, you can get significantly different frequencies. So the effect sizes here aren't huge. I'll show you some more compelling ones here in 1 second. But at low altitudes and high altitudes, at the same time, the same day we sample, Wolbachia prevalence can vary. And that starts to imply that maybe there's some environmental effects. We see larger patterns. So if we look across continents, this is for WMEL in Drosophila melanogaster. The piece of the pie that's gray here is how common WMEL is in these Drosophila melanogaster populations. And this is work from Ari Hoffman's lab that we've been building on recently. What you can see is that in the tropics, WMEL is pretty common. In the temperate zone, it's relatively uncommon. And you see this pattern of clinal variation. Notably, that's not restricted to Australia. We see a similar pattern in North America, where frequencies tend to be higher at lower latitudes here, which are also subtropical in this case, in the case of southern Florida. So what might actually govern how common Wolbachia are? And I think there are two things, at least that my group is really focused on, that we know are important for this. And a lot of this stems off of theoretical work that has identified these as the most plausible candidates, work carried out by Michael Torelli and his colleagues doing theory work on this. And those two things are variable CI strength and variable Wolbachia transmission rates. So we often present CI in this context of, if you're an uninfected female and you mate with an infected male, your eggs don't hatch. But this can vary a lot. Okay, so for example, if we look across systems, what I'm showing you here, and this is the work of a wonderful postdoc in my lab, Dylan Shropshire, who actually did his beautiful PhD work with Bordenstein lab from the talk the week before. You saw other work from their lab. Dylan did his PhD work there. He's now in my lab trying to understand why CI strength varies so much. But to convince you that it does vary, here we have a bunch of different Wolbachia variants on the x-axis that infect various Drosophila species in their natural background. On the y-axis, we have eggs hatched, and I'm showing you both compatible crosses and the CI cross. So a signature of CI is when the CI cross, the bar is very low proportion of eggs hatched. So if we look at WRI, the one I already told you causes strong CI, to get to high frequencies across the Australian continent, like I showed you in that original plot, you can see that it causes very strong CI. Indeed, Dylan has given us the range here. So if you just want to look at sort of the percentages, you can see on top where I'm highlighting here. The point I want to make is that it can vary a lot. So let's look at WMel, which is interesting because it causes very strong, if not usually complete CI in Aedes aegypti. Under the conditions we do here, it caused relatively weak significant CI. And this is a pattern that's typical for WML. If you use very young males, you can get very strong CI. But if you were to cross, say, 5-day-old males under typical conditions, you tend to get relatively weak CI. Drosophila tessieri, in this experiment we got, and this is a WML like Wolbachia, and this is a Drosophila yakuba clade fly, in the same clade as yakuba and santomea. You can see reductions in the egg hatch from the CI cross that are non-significant. I had previously shown in 2017 that you can get CI for a WT cross, but if you then introgress that same variant into a different tessieri background, you sometimes won't get CI. So that suggests that host backgrounds are modifying CI. And that makes sense because theoretically, we know that hosts should evolve to suppress CI, which I'm happy to talk about later. The point is Dylan's work and work of others is really showing the extent to which CI strength can vary. Again, also, CI strength can vary within system as a function of things like male age and other factors. Before I move on, I do want to plug that Dylan, he's a wonderful postdoc, and he's starting his lab at Lehigh University this fall. If you're interested in any of this CI work, I would urge you to reach out to Dylan. He's a wonderful mentor doing some of the best science in the field. And like I said, he did his PhD in the Bordenstein lab and his postdoc here. So he has what I consider to be a pretty unique skill set going from mechanism to population biology at this point. So what are the causes of this CI strength variation? I can't totally answer this question, but what I can do is convince you that it's complicated. So the way we think about this is the possible genetic contributions, and the way this system is outlined is that we have hosts. We have host genomes that can vary. We have Wolbachia genomes, obviously, that can vary. Inside those, you learned last week, are prophages that are living inside of Wolbachia genomes. We have the CI loci that Dr. Carr introduced last week that are associated with these phage regions. So you can think about CI loci or inside phage or inside Wolbachia that are manipulating host reproduction in this case. We've shown in some of our systems, and we continue to work on this, that it can even be a little bit more complex. So sometimes CI loci are flanked by transposable elements that can lead to CI loci themselves moving horizontally among very closely related Wolbachia. So you can have CI loci flanked by TEs that can encourage their horizontal movement. Those are in phage, which could presumably also move horizontally. We have Wolbachia that are moving horizontally among hosts. And this entire system here is leading to manipulations of host reproduction that are pretty dramatic. So I think this is why this system is so interesting to us and obviously other people. So let's think about the genetic contributions, and just let me convince you that there are multiple things that can contribute to CI strength variation. So Dylan's doing some really beautiful work now. This is preliminary, but that the strength of CI caused by any particular variant can depend on host background. Like I said, I had results in the yakuba clade in 2017 showing we could sort of turn on and off CI as a function of host factors that are unknown. Dylan's interested in better understanding those. And some of this work goes back to results from Ari Hoffman in the early 2000s, where he's observed some of this in the WML melanogaster system. What about the broader Wolbachia genome outside of cifs? Well, you could imagine if there are loci that contribute to things like Wolbachia replication and how abundant they are in host tissues, that could matter. It doesn't explain a lot of variation, but Dylan has observed that CI strength tends to be higher when Wolbachia are particularly abundant in male testes in systems he's looked at. I'm going to skip phage and TEs for this part because I just wanted to sort of introduce those in terms of the nested system. But obviously, CI loci, you learned last week, have dramatic contributions. Those were discovered by really beautiful work in the Bordenstein lab and John Beckman's lab in 2017. And Dylan contributed to the discovery of both those and the rescue factors. He has a really nice PNAS paper if you're interested in the discovery of the rescue factors. He's now going on to characterize the diversity and structure and function of CI loci. We originally thought, or at least it seemed like, that you had these different types, type 1s and type 4s, that are deubiquitolases or nucleases. As you learned last week, it's more complex with that, where they can have multiple functions. Dylan is looking across many different cifs to try and characterize the variation that we observe in there. And you can see that there are cifs that are very closely related, that are very divergent, that have similar or different structure. And all of these things can contribute to CI strength variation. If you're interested in this, stay tuned for more stuff from Dylan as he starts his own lab. One of the things I'm most excited about that Dylan recently did, that's been published in the last couple of months in PNAS Nexus, is he looked and was trying to figure out, what might be better predictors of CI strength variation. And so he looked across a bunch of different Drosophila variants, including WML and WRI, and then a bunch of these that are from clades that we've been developing in my lab for a while. If you're interested in any of the particular ones, just let me know. But what you can see is he has these characterized as if they cause relatively weak CI or relatively strong CI or no CI. And then what he's plotting here is the expression of one of the cif genes, in this case cifB. And what you can see is that cifB expression tends to be a good predictor of how strong CI is. So variants where we get no CI, and I should say by no CI, I mean not statistically significant CI. When you look at the crosses, you tend to see non-significant reductions in egg hatch, for example, for his most recent WT crosses. But again, we have other crosses where it is significant, but it's always very weak. You tend to see, in this case, low cif expression. In the case of those that cause weak CI or intermediate levels of CI, you tend to see a little higher cifB expression. And then the highest cifB expression Dylan sees for those variants that cause the strongest CI. Now, while this is a good predictor, as with most things in biology, and particularly Wolbachia, it is complex. So we do have variants that cause very strong CI where cifB is expressed at very low levels. And Dylan is working on resolving this, and it could very well have things, have something to do with, for example, the localization of interacting products. And so stay tuned again for that if you're interested. But on average, I think cifB transcription is a pretty good predictor of CI strength variation. I'll just briefly show you one other result from the last few years in the lab that's pretty exciting. So what about single nucleotides inside cifs? So now if we're looking, at this sort of nested systems, what about individual nucleotides that are in there? What Kelly Van Berenberg in my lab and Daniel Aqua, a master's student in John Beckman's lab, did is they took the WML, the typical WML cif, then the discovery of those being cifs was mostly transgenic CI expression. They took that and they also expressed that transgenically in a Drosophila melanogaster background. But what they did is they introduced mutations, they expressed them with particular mutations that we observe in sister Wolbachia. So in the Drosophila yakuba clade, we have these Wolbachia that are very recently diverged. There are very few mutations that separate them. And so they've expressed these. We have an uninfected by uninfected cross. We have one that has the typical WML construct being expressed. And then we have one that's expressing one with a particular mutation that changes a valine to a leucine in the cif. And what that ends up doing is reducing the strength of CI by about half. And then we showed that has to do with reductions of enzyme activity and inefficiency, reductions in those, particularly the DUB region, the deubiquitinating region of this cif. So even single nucleotides can affect this. So what are the causes of CI strength variation? Again, it's complex, but contributions of the host genome, the broader Wolbachia genome, things related to titer and how many Wolbachia there are and where they are, cifB expression, and then obviously CIFs generally, but even individual mutations in there can affect CI strength. Importantly, Dylan has really beautiful work on host age. We're doing work on temperature stuff, building off some other work, and these things can all influence how strong CI is. So it's complex, but just know their CI strength varies greatly, and there are a lot of contributing factors. This is important, remember, because I've told you very strong CI leads to high Wolbachia prevalence in nature. One thing I haven't really hinted on is that when CI is relatively weak, Wolbachia tend to occur at lower frequencies, usually intermediate frequencies that vary. So those data that I showed you for Drosophila yakuba, where Wolbachia prevalence is varying through time and space, those are relatively weak CI causing. You notice that Wolbachia tend to occur at intermediate frequencies, but they also vary. So CI strength contributes to that variation. But remember, I told you there's one more thing that I think could be important for governing Wolbachia infection frequencies in nature. And I want to show you some of our recent results that suggest it could be really important in a particular system that most of us care a lot about, and that's WMel and melanogaster. So I want to bounce back to this result, and this is produced in Australia here from Ari Hoffman's group, remember, showing that WMel frequency is quite common in Drosophila melanogaster populations in the tropics, relatively lower in the temperate zone, and that tends to be equally true in North America. So we have these parallel clines in Wolbachia frequency, WMel frequency. This work is led by a wonderful postdoc in my lab, Mike Haig, who's on the job market. So if you see him popping up in a pile for your positions, this is his work, and he's an excellent colleague that I hope you'll consider. He did this work, or much of the work I'm about to show, with undergraduate Chelsea Caldwell and a newer graduate student, John Stotts. So what we wanted to do, what I thought would be a relatively simple hypothesis to test is it could it just be temperature that's involved in this pattern, right? Because if we know what varies with latitude, temperature is something that really varies. So what we did is we reared flies in different temperatures, and then we measured how well they are at getting Wolbachia into the next generation. Okay, and so what you're going to see for all this are genotypes on the x-axis and maternal transmission rates on the y. When it's very high, that means all of the offspring or most of the offspring are getting Wolbachia. Okay, on the x-axis, we have both temperate genotypes. So those are Drosophila melanogaster genotypes sampled from the temperate zone infected with WML. Tropical genotypes, those are genotypes sampled from the tropics infected with tropical WML. And then we have each of those genotypes where we've reciprocally integressed them. So temp trop here in superscript would be a temperate Drosophila melanogaster with a tropical WML Wolbachia and vice versa. And this enables us to look at interactions between host and Wolbachia genomes, and in addition to looking at the temperature effects. When we rear them at a typical 25, we see a similar pattern. We see a little bit lower transmission rates of Wolbachia, but when the pattern was really revealed is when we reared them at 20C, a temperature that's representative of the temperate zone down here, where transmission really starts to break down. So these are the same genotypes. The only thing that's happening is they're being reared at relatively warm tropical-like conditions, intermediate, or these cooler conditions. One interesting thing you can see is that at a cool temperature, you particularly get breakdown of transmission with the tropical genotypes that have evolved in these presumably warmer climates. And the differences that you're seeing here leads to an overall G by E interaction, or an interaction between host genome, Wolbachia genome, and temperature. Okay, so transmission is breaking down, and it's breaking down in cool environments that are representative of the temperate zone. What we next wanted to do, though, is ask this question: could this explain WML prevalence across the cline? And so we used models developed by Michael Torelli. What I'm showing you here are the gray bars are the observed frequencies. So in Melbourne, this is the observed frequency of WML in host populations. The dotted line is the mean. Same thing up here, observed frequencies. On the y-axis, we have the equilibrium frequencies. So this is from the model, the predicted equilibrium frequencies. And then on the x, we have relative fitness. So we're doing this across a range of plausible fitness effects of Wolbachia, which I can talk about here in a second. When we do that, what we see is regardless of how Wolbachia affect host fitness, the models suggest that if we only consider temperature effects on transmission, we can do a great job of explaining how common Wolbachia are. The same is true in the temperate zone, with the only difference being that we need to assume subtle positive fitness effects. I hinted at this at the beginning, but we know from theory and we know from empirical observation that to initially spread, Wolbachia must positively increase host fitness. This level that we're assuming to be able to explain their frequencies here is well within that plausible range, given what we know. So in total, we think we can provide a pretty simple explanation for continent-wide variation in how common Wolbachia are if we just consider how good mom is at getting Wolbachia to their offspring as a function of temperature. Notably here, we need not even really think about CI. So we're just assuming extremely weak CI. We don't think about CI strength variation. It turns out CI is not that important in the system. You might be wondering, what's the cellular basis of this? So what we did is we looked at oocytes, developing female oocytes under the same conditions at these stages where Wolbachia need to get to the posterior pole to get into the next generation, to get into the germline. We did this at 28. We did this at 25. I'm summarizing, these are just focal images, and I'm summarizing the amount of Wolbachia in these various regions, the whole oocyte, the posterior region, and then the pole where they need to be to get into the germline. You can see 28 and 25 are pretty similar, just like transmission. But when we do it at 20C, what we can see is we get significant reductions in the total amount of Wolbachia in the oocyte, the amount of Wolbachia that are at the posterior region, and the amount of Wolbachia that are at the pole where they need to be. As I noted, we had G by E interactions in the transmission data, and so we're interested to see if there's any evidence of local adaptation here, differences between the temperate and the tropical genotypes. What we see is that when we rear them at cool temperatures, W-mel from the tropics and a tropical melanogaster have lower oocyte abundance in the cold, and they have lower Wolbachia abundance at the posterior region and lower Wolbachia abundance at the pole. So this is evidence that if you're sampled, if you're evolved in the tropics, we sample you from there, you're particularly negatively influenced by cold temperature when it comes to transmitting Wolbachia. This is interesting because these Wolbachia diverged only about 2,000 years ago, so they're very similar. I'm just showing you a tree of W-mel here, a Bayesian phylogeny, with W-yak, this closely related variant, as the outgroup. But our tropical and temperate variants are really closely related. Sadly, we can't genetically modify Wolbachia, which I think anyone in this series would agree. Would agree that we wish we could do that. But what we can do is genomics, and so I'll show you what we've done and as close as we're able to get to this so far. In terms of the genetic basis, we have a 1.25 megabase genome. Interestingly, among these temperate and tropical lines, there's only 43 SNPs distinguishing temp and tropical. Only 20 of those are non-synonymous. Only 13 of those are ones we would consider to be non-conservative. And only one really makes sense in terms of host Wolbachia interactions, and that's this Wolbachia surface protein B. When we look at that, what we see is a stop codon. This is disruptive, we think likely pseudogenizing stop codon, and we observe it in the tropical variant. This is an outer membrane protein likely involved in communicating with the host, so you can think about this being important, for example, in the process of getting to where you need to be to get into the next generation. We obviously don't know yet exactly what's going on, but this is, we think, a very good candidate so far. Interestingly, this is a derived stop codon. We only observe it in W-mel. We've looked across 50 million years of divergence in Wolbachia genomes, and we only observe this mutation in tropical W-mel. We wanted to take it a little bit farther to see if we could get any better evidence, to see if this is something that's involved with this breakdown of transmission as a function of temperature. So we looked at global population genomic variation. We looked at 69 localities, 109 population genomic samples, and about 20,000 SNPs. This is a beautiful data set, this DEST data set that you can read about in Compute et al. 2021, a really nice resource for the community that we were able to pull Wolbachia reads out of. And what we're interested in is, do we see patterns of genetic variation in W-mel co-varying with temperature globally? What we see is that 8 of the SNPs distinguishing tropical and temperate W-mel genotypes variants fall in the top 1% of all temperature-associated SNPs globally, including 4 of the top 5, with WSPB being the third strongest association with temperature. So there are some, we can't genetically modify Wolbachia, but Mike is really interested in figuring out what WSPB is interacting with on the host side. Like, we think we've done a good job of explaining how temperature is affecting abundance of Wolbachia oocytes and how that ultimately influences prevalence. And we think we have a good hypothesis here that Mike's interested working on in terms of what the genetic basis is. But again, we consider this to be a hypothesis at this stage. In summary, temperature effects on cellular Wolbachia transmission seem to provide a simple explanation for global variation in WML prevalence. I think this is interesting. For those that know, in the 80 systems, there's also evidence of temperature effects that can affect transmission, although those tend to be related to heat stress. We could potentially talk about that if people are interested. A larger summary, Wolbachia are naturally and rapidly moving non-sexually among hosts diverged more than Drosophila and 80s are diverged. I think that's interesting, and understanding the mechanisms of that is something we're really interested in. CI strength varies widely for many reasons: the host genome, the Wolbachia genome, cifs, even individual nucleotides, male age, temperature, lots of other things. That's very important because CI strength, particularly when strong, can drive Wolbachia to high frequencies. And then finally, as I just showed, we think temperature effects on maternal transmission are particularly important, because if we know that, if we can figure out how much maternal transmission is being disrupted using the mathematical models, we've been able to see that can give us a really simple explanation for whether Wolbachia are common or not in host populations on continent-wide scales. I've tried to give credit throughout to all the wonderful people in my lab that have contributed to this, but there are many others. I've been very fortunate to have so many talented junior trainees in my lab, and I just want to thank all of them. I'd also like to thank my funding. I've been very fortunate with that, so I'd like to thank the NIH and the NSF. And then I have several collaborators that have been particularly important for this talk: Michael Torelli, Ari Hoffman, Daniel Matuti, Bill Sullivan, Kim Stanek, and then John Beckman contributed to work that I highlighted here. And with that, I would take any questions. Thank you for your time.
Great. Thanks a lot, Brandon, for a wonderful presentation, and with some really interesting biology that I certainly wasn't that aware of. I'm going to invite the audience to ask questions. You can do that in two ways. One, you can either chat your question in the chat feature, or if you would prefer, you can raise your hand and we will unmute you and you can ask your question directly. So please go ahead and do that. Brandon, I want, if you could talk a little bit about the temperature correlation that you saw, that the clinal variation in frequency that you saw seemed to be opposite of what is seen with WML in Aedes aegypti, and you touched on that a little bit at the end, as perhaps it was there's a different phenomena going on there. But in Aedes, it's in high temperatures we see lower WML transmission. Is that accurate? And can you comment on that?
Yeah, sure. I'd be happy to, Dave. And thanks again. Yeah, so Perrin Ross and Ari Hoffman and others have worked on this, and it's true that heat stress can have effects on both CI and transmission in Aedes aegypti. I think it's currently unclear if these are fundamentally different responses in the different systems. And I say that because we haven't looked at heat stress. They were looking particularly, I think, if I recall, at cyclical heat stress. I'm forgetting about the difference between the CI experiment and the maternal transmission, but point is heat stress. We've looked at 28-degree developmental environment, which for melanogaster is not that stressful for the genotypes that we're using. And they obviously haven't looked at cool temperatures. So it's totally plausible that the results would be relatively similar if we were looking from, say, across the full range from cold stress to heat stress, and there's just an intermediate environment that tends to favor transmission. So I think we don't know that yet, but you're absolutely right. What we do know is that cold temperature negatively affects transmission in Drosophila in the natural system, heat in the Aedes system. Notably, though, there are interesting differences for other phenotypes, right? Because I've said that, and I showed you that WML CI strength is relatively weak in Drosophila melanogaster unless you use very young males, whereas it tends to be very strong or complete in Aedes aegypti. So this could also be things related to host effects, just like it probably is for CI as well. And I think we just need to do more experiments to answer your question.
Yeah. Yeah. Is this these phenomena that you're seeing with temperature, is this is this seen also with other Wolbachia variants? Not just, most of your work was with W. mel. Would you see this with other species or strains of Wolbachia as well?
Absolutely. So that's a great question and something we're doing right now. So we're extending this work in two ways. We have a paper that we'll post on bioRxiv probably later this week with Bill Sullivan that's looking at the cell biology in a single temperature for all these variants. And so those oocyte patterns that have been restricted in terms of analysis to W. mel and WRI, we're now extending those across Drosophila, and that will show that there are quite different strategies for getting into the next generation, I think, or at least the mechanisms imply that that's true. And then in my lab, Mike Haig is extending his results and those that I just mentioned to different temperatures. And so Mike is currently measuring transmission across different temperatures for a bunch of different Wolbachia variants. And then also at the same time looking at how those cellular distributions differ in the cold across those. So I can't answer it yet. I can tell you that preliminary data suggests there are other systems where this happens just like this, and there are other systems where they seem to be far less sensitive to temperature perturbation. Obviously, we're interested in this because I suspect that this will co-vary with how stable infections are in nature. So we're interested in testing the hypothesis that how sensitive they are, say, to temperature, and the transmission breakdown is a function, or at least is predictive in terms of how variable Wolbachia frequencies are in nature, right? Where those that are stable tend to be less affected by abiotic conditions, for example.
Mm-hmm. Yeah, let me encourage our audience to go ahead and ask some questions. And we'll be happy to address them. You can raise your hand if you'd like. We'll try to recognize that as soon as you do that, and we can answer them. One of the things that I found that to be really interesting was the first part of your talk, where you didn't have a lot of answers, but you had a lot of interesting phenomena, which was the horizontal transfer of these things and the widespread horizontal transfer of these Wolbachia. So, I mean, can you talk a little bit more about possible mechanisms, and is there some preferred mechanisms that people think are more likely than others?
Yeah, I think the literature, if you were to look at the literature, there are a couple that I think are plausible that people think would be supported. One is parasitoid wasp. Interactions with parasitoid wasps, which could make sense. I think the question there would be is are they sort of moving Wolbachia around between different infected species, or are they moving their Wolbachia into different species? Another would be there's some papers out there suggesting that for things that associate with plants, that you could have interactions with plants that lead to the possibility of horizontal transfer from future individuals that interact with those plants in similar ways. But I think there's a lot of questions about how well Wolbachia can do, say, outside of the cell. How is this happening? The way I think about it on average, Dave, is that there's probably a lot of different species interactions where this can happen. I think about it sort of like mutation. Every generation, mutations are falling, and we only sample some of those that tend to not be too deleterious, right? In this case, I think you're getting these interactions with the possibility of horizontal transfer happening all the time, but it only takes off sometimes, right? So it has to be a Wolbachia variant finding itself in a host background, probably needs the starting titer that gets donated, needs to be enough to where it can hang around and persist, and then ultimately get into the germline.
It's got to get into the germline, right? So I mean,
we know this can happen. I mean, I can inject adults in my lab and get it to where it goes vertical. It's much harder, though, to do that and to come back, say, in 10 generations and it still be there, right? So this process can happen. Horatio Friedman has interesting papers people could look at from Boston University. on the actual mechanisms of Wolbachia getting into the germline.
So you can inject just Wolbachia, extracellular Wolbachia into adult hemolymph, and you can get transmission.
You can spin, basically really coarsely spin Wolbachia out of, through a filtration process, or you could pull them out of a donor egg, inject them into the soma, and yes, get Wolbachia moving into the next generation. Again, we've done this and had very little success with getting an established genotype across many generations, but we have had a lot of success with getting it into, say, F1s and sometimes F2s.
Yeah, great. Let's go to a couple of questions that have come in while we were chatting. Let's see. First question by Nicholas Road. Hi. Is there evidence of uninfected females discriminating from uninfected males? So I guess sexual selection here.
Yeah, so the idea that if uninfected females could sort of sniff out whether or not you have an infection, that would be a very good thing, right? Is that the question, where you preferentially mate with uninfected males?
Yeah.
Yeah, so some evidence of that in the literature, but I would say I don't think that this is a super common thing, because we go out into nature and we see so much evidence of CI, right? And that this, selection on that should be relatively strong, I think. I was interested in the past of looking, so in insects, they communicate through things like cuticular hydrocarbons. And so I think a question that would be interesting, and I haven't looked for a while, so I'm sorry if there's someone here elsewhere that has done this and I don't know their paper, but I think it would be interesting to know how Wolbachia maybe manipulate things like cuticular hydrocarbons, because that would be the mechanism, I think, of the way that would happen. And then Wolfgang Miller's lab has some really nice results in Drosophila polystorum on how Wolbachia might be related to things like pre-mating isolation. So I would direct people to the Miller lab's very nice work on those sorts of things. But we do know, as you can see in this picture, that Wolbachia are in the brain. We do know that they can co-vary with behavioral phenotypes, and so I think it's a great question.
Yeah, great. Stephanie James has a question. It says, it wasn't clear to me whether use of the tropical variant of WML from Drosophila might overcome the heat sensitivity observed with WML and Aedes aegypti. Yeah, so back to this question of mosquitoes.
Right. Yeah, Stephanie, I think that's a wonderful question. So I haven't spoken with Scott O'Neill or others recently to know how the sort of selection process for the variants that are actually used is happening. I think initially it was just, here's a WML variant, let's put it in there. Does it work? That sort of thing. I know they're definitely must be thinking about these things now. I think, we don't know if our tropical variants are more heat tolerant in melanogaster, and I certainly don't know if our tropical WML variants are more heat tolerant in Aedes aegypti, but I think that is exactly what these results, to me at least, suggests we should do, is that we should start thinking about, and I don't mean to imply that people doing this work aren't already thinking about it or weren't before this, but I think these data definitely suggest that temperature is interacting. It's definitely interacting with Wolbachia and the host genome. There are these three-way interactions. And so I definitely think there are Wolbachia WML variants that you could sample and use for these approaches that would be better than others. But I don't have any specific data for tropical temperate variants to justify that, just these data from the natural system that motivate that. Yeah.
So some of Steve Sinkin's work and Tom Ant's work, they've been working with alb-B, and they've shown that the temperature, it has different temperature sensitivities, or it's much more temperature tolerant in infected Aedes.
Yeah, exactly. I think so that is clear. It's clear that we see this in this system, but I think a way forward, at least for us, because we're working in natural systems, is to think about how these behave in the natural systems and if that sort of maps onto the way they behave in the others. And so we have some collaborations with people ongoing that are aimed directly at figuring these questions out. So that's another great question.
Dorrington Agoye, Dr. Dorrington Agoye asks, thanks for a very informative presentation. Could you comment on how the CI strength has been optimized in some of the mosquito population replacement programs that have been launched?
Yeah, That's a great question. The reality is that when you take WML and you put it into Aedes aegypti, it causes stronger CI, and indeed, under most conditions, complete CI. So how do we think about that? Well, the way I think about it is that we know natural selection should or should favor suppression of CI, right? And so in this case, you're pulling WML out of its natural host, you put it into a novel host, and you get really strong CI. I think that the most plausible explanation there is that you have host factors that in melanogaster that are modulating the strength of CI. We're actively right now looking for those. I can't tell you anything specific on it, but I will be able to tell you soon about these host suppressors. So I think there's host suppressors that have evolved in natural systems. You break up those combinations in natural systems. In a novel background, you don't have that, and you get strong CI. An obvious question is what is the time scale of the evolution of suppression? Because I think a question that would bounce off what I just said would be, okay, well, are suppressors going to evolve in Aedes aegypti? The answer is yes, I think, conceptually, but the time scale of that is sort of challenging. It's similar to this questions. I sort of proposed some things that I wanted to talk about and didn't have time. We know that there's weak purifying selection on CI, so we actually expect that CI will break. There's evidence of that in natural systems that we've shown. This is based off of theoretical work by Michael Torelli. Luckily, the time scale of that, at least from what I've been able to figure out, seems to be many hundreds to about 1,000 years. So I don't think that mutations are going to break CI and mess up the efforts that are going on now. But that whole process of CI's effects being frequency dependent is why that's true. So remember, I said when Wolbachia first get into a system, CI doesn't really matter because uninfected females are mating predominantly with uninfected males, just probabilistically because they're more common. And so that leads to weak purifying selection and mutations in CI that ultimately we expect to reduce CI strength. And we see that in systems like, for example, a B group variant called WMA that infects Drosophila mauritiana is one system where we've worked on that.
Mm-hmm. Tom Wolf has a question. Is Wolbachia better thought of as a population of individuals with different variants or as a strain with fixed effects?
So that's interesting. Yeah. For cataloging purposes, we call things, right? So we say WML because that's a thing that's sampled from WML. But I can tell you right now, I can go sample something that is effectively WML in a different species that's very closely related. For example, the yakuba clade, it's diverged a few thousand years. We call it WYAC because it's sampled from there, but that is effectively a WML variant, a WML-like variant. So there's a question about how differentiated things need to be for us to call it the same thing. It's absolutely true if you go out and you sequence WML from a bunch of Drosophila melanogaster individuals, there's genetic variation there. So I can't, we tend to call these WML just because they're from melanogaster, but there is genetic variation there, and I definitely think about it as a population. I think about it in terms of a population, just like I do with the insects. Of course, it's more complex because you can also have heteroplasmy, like you would with mitochondria, for example, potentially with Wolbachia, meaning that within hosts, there could be variation in WML within the individual. And then we also have systems where there are double infections where they actually have two different Wolbachia variants, right? Yeah.
Yeah, so Tom has a follow-up here. It says, to what extent do Wolbachia positive effects versus reproductive manipulation effects have a role in the spread of Wolbachia? And are there any indications that we quantify one effect versus the other?
Yeah, that's a great question, Thomas. So there are three parameters we have to think about that have been identified in the modeling efforts over the last 30 years from Michael Torelli and Nick Barton and other people that have worked on that. And that is the relative fitness effects of Wolbachia on hosts, reproductive manipulations on hosts, and imperfect transmission. When we think about how common they are, you can think about both fitness effects and reproductive manipulations, pushing Wolbachia towards fixation. Right? Because if you're in the case of CI, if you're infected with Wolbachia, you can mate with both classes of individuals. So once you become relatively common, CI becomes really important. To get there, though, you have to get there other ways. So this is what we do know, Thomas. We know that positive fitness effects are required. They're necessary to spread from low frequencies. CI cannot spread you from low frequencies. We have evidence that in some systems, those positive effects are related to things like fecundity. There's some evidence of, say, virus blocking in natural systems that could provide a relative fitness benefit. And so it's a classic problem that evolutionary biologists thinking about fitness regularly get into. There, are 1,000, 1,000,000 components of fitness, depending on how you break them down. We could measure many things. Some of those things we see, do correspond to increases in fitness, but we can't measure everything. So it's a theoretical argument that Wolbachia that we sample in nature must positively affect host fitness to initially spread from low frequencies. We have an idea about what some of those things are. It's a theoretical fact that CI does nothing at low frequencies to spread. But when you get to that tipping point equilibrium where those mostly unknown positive fitness effects get you to appreciable frequencies, where males are now common enough that are infected to start to encounter uninfected females, then CI really pushes you to high frequencies. Then it's really kind of all about CI rapidly pushing you to high frequencies. And then whether that ultimate prevalence is 80% or 90% or 99% is a direct function of how good mom is at getting Wolbachia into the next generation. So in WRI, mom is pretty good at getting Wolbachia into the next generation. It seems to be less temperature specific, for example, in some of our preliminary data I didn't show today, whereas WMel causes relatively weaker CI and also tends to be, at least in our preliminary analyses, comparing it to WRI, tends to be more temperature sensitive. So all of those things interact to do it, and the equilibrium frequencies are ultimately balanced by those reproductive effects, the fitness effects pushing in one direction, and the every generation uninfected individuals being inserted back into the population due to imperfect transmission that effectively reduces prevalence. Hope that answers your question. There's no exact number. This is the exact contribution of fitness effects beyond they're very important when Wolbachia are initially rare.
Good. I see we have somebody raise their hand. I'm gonna recognize them. Bilal Rasul. I'm going to unmute you, Bilal, and why don't you go ahead and ask your question? And you'll have to unmute yourself, I think. Bilal, are you there?
I think we lost Bilal.
Okay, yeah, I guess we did.
Good comment in the chat, Bilal. If you have a question, I'd be happy to answer there.
Yeah. Brendan, I wonder if you can go back and fill in something I really missed a little bit of, maybe the punchline. It had to do with the WSPB protein.
Yep.
That could you just sort of back up and just sort of maybe rewind that a little bit and go through that again just for my my benefit at least?
Yeah, absolutely. So what we know is that temperature affects the abundance of Wolbachia in developing host oocytes, and in particular, the abundance of Wolbachia where it needs to be to get into the germline at a particular stage of oogenesis. Okay, so temperature, cold temperatures in particular, affect that. So the cellular basis is pretty clear. Wolbachia abundance and localization matter, temperature interacts with it, and that breaks down transmission. We're interested in understanding, though, what the genetic basis of that might be. I don't think that you have any confusion. It's just that we can't genetically manipulate these, so all we can do are present, correlative genomics results, right? That's an inherent problem in the field, And so what we see is we see these different patterns, for example, at the cellular level of when you raise infected hosts in cool temperatures. Tropical variants, for example, have lower Wolbachia abundance at the whole oocyte, the posterior pole, the posterior region, and at the pole, where they need to be to get into the next generation. And this is the exact environment where transmission breaks down. So there's just fewer Wolbachia where they need to be to get into the next generation. What's surprising is these differences between the tropical and the temperate variants is that they're nearly identical, right? I mean, these are WML, back to that question that I think Thomas had. These are they're WML but there is genetic variation and they differ, right? But they're very closely related. And so then when we just look, we say, okay, well, how closely related are they? Can we do some genomics to try and figure out what things that differ between these two variants might matter? And there's so few variants, we can start to whittle it away. I'm not showing you every single thing that varies, but basically we get it down to 13, and there's just nothing that makes sense for interacting with hosts in a way in which could affect transmission other than this surface protein, Wolbachia surface protein B. And so WSP, another Wolbachia surface protein, is sort of well known because it's been used to type Wolbachia for a long time. This relatively conserved locus, that you can use to type Wolbachia in PCR assays and stuff. This WSPB, what do we see in this when we look? We see a stop codon, right? And so not only are there very few differences among our temperate and tropical WMel, there's only one serious mutation. It happens to be in WSPB. It happens to be a stop codon. And this happens to be an outer membrane protein that we know should be involved in communicating with those cells. Now, the reason I can't give you a punchline is because I can't genetically manipulate it to explicitly test it. But some of the things we can do that Mike's interested in is identifying what it interacts with on the host side and then genetically manipulating those. And so that's what Mike is doing now, and that's what would give you the punch. But I think it's interesting that we only see this in WML, right? You look at, it's a derived stop codon. You do not see this in any of these other things across 50 million years. And then it co-varies with temperature globally, right? And so when you look globally, you say like which SNPs, what genetic variation co-vary with temperature, it's the third thing with the strongest covariance in terms of its frequency in host populations with temperature. And so there's all this anecdotal evidence, right? If we could nail this down, I think we would have been able to publish this in one of the most high impact journals, but we're in a journal I know and love at Current Biology here, explicitly because we figured out everything except the genetic basis, right?
Yeah, speaking of the genetic manipulation, I mean, are you optimistic about the possibilities of doing that? I mean, people are now, genetically manipulating microbiome components that are unculturable, using various approaches. And of course, there's a lot of interest in manipulating microbiomes, particularly from the health perspective. And people are getting very creative in how they can go into microbiomes at least and manipulate them without having to culture them.
Absolutely, yeah. So I tend to be optimistic and think we can do anything. So I'm the kind of person that will say, yeah, I think this is not an impossible thing. I don't think we're going to do it. We actually tried early on some, I think many people in the field have, culturing Wolbachia under different conditions I did it with microbiology colleagues that have worked on this with other things. People that have much better skills in this arena than I do. I think, Jason Raskin in the field, I think back in the days worked on this, others have. And if you talk to people, they will all tell you it's very hard. We've tried everything we can think of. And so I guess my larger answer is I don't think I'm the best person to ask because I know I won't be the person to do it. But I think absolutely, given some of the data we see from other systems, I'm cautiously optimistic that there might be ways forward. But yeah, I'm pretty confident that we won't be the lab that does that. We're not working on that.
Yeah, okay. Hey, Tom Wolf has come back with so could CI be considered as a bet hedging strategy that kicks in only after a certain frequency is in the population, maybe involved in dealing with different host environments, new tissues, new cellular environments, et cetera?
Yeah, another great question, Thomas. I don't think about it that way because CI itself is operating the same way. And any focal cross is operating that sort of the same way in a system. What I mean is that uninfected female mates with an infected male. You get CI at the population level. It's just not sort of kicking in to drive it just simply because uninfected females aren't encountering infected males until it already becomes relatively common. I don't know if that directly answers your question. I don't think about it that way, but I know why you're asking it in that way. But CI is, all the contributions are complex. The strength can vary among environments, genomes, and all these other things. But on average, whether it matters for spread, if that's what your question is related to, it's literally just a function of, has it become appreciably common or not, such that uninfected females with some probability mate with infected males. Yeah.
Yeah, good. Oh, my question about WSP and asking you to go back has generated a comment from Julian Martinez. It says, nice talk. WSP is also a candidate gene among WALB variants in transfected Aedes aegypti that differ in heat sensitivity. It's pseudogenized in the more heat-stable WALB variant.
Very interesting, Julian. I don't know if this is something you're working on, but I'd be excited to talk to you about it. I love your recent work. Your recent work with Jiggins on CI and other stuff. Very beautiful work. I'd be excited to talk more about this. Mike Haig in my lab has taken the lead on this, and I'm sure Mike would have a better answer than I do, but that's extremely interesting and love to talk to you more about it. We might send you an email on that. Cool. Preprints out. All right, great. It's probably in my list and not read yet, but I'll take a look at that and then maybe shoot you a note, Julian.
Yeah, he says the preprint is out.
Yeah, perfect.
Okay, so thanks for everybody else who didn't see that comment. Well, great. I'm going to, before we wrap up here, I'm going to just say one thing. Let me just share my screen. And hopefully people can see that. So I just wanted to say, first of all, thanks a lot, Brandon, for a really interesting webinar that really fits in really fantastically with the rest of the series. And I wanted to tell everyone that the series will continue. We have 2 more webinars within this series. There will not be a webinar next week in the series, but the next webinar in this series will be on November 9th, and Julie Dunning-Hottop will be speaking on the topic of basically some Wolbachia genomics and some of the work that she's done, where she's looked at gene transfer from Wolbachia to the host genomes and those kinds of things. So I encourage everybody to come back on November 9th for that webinar. So again, Brandon, thanks again for taking the time this morning to speak with us, and thanks to the audience who attended and for those who asked questions. So thanks a lot. And at this point, I'm going to close our webinar.
Yeah, thanks everybody. Really appreciate the opportunity, and it's great to talk to everybody. Send me a note if you have any questions.
Yeah, great. Thanks again.
Thank you.